Okay, today I'm going to present the second lecture. This lecture's content is about Channelrhodopsin-dependent green algae behavior. So today I'm going to talk about generally about this model organism, the green algae, the climate dominance, rain-hardy, and the structure of the eye spots, and the movement that's because of the photo response. So first I will try to label it with highlight. No, so not this one. Join spotlight. Let me see. Annotate. Join mouse. Okay, didn't see that. Hopefully have this arrow or spotlight. Okay, now I got the spotlight. So can I move this table to here. Then I will try this spotlight. So there's a no. And let me go back to see. Okay, now let's go back. Still give this annotation and the spotlight here. I will try to remove this, the whole panel somewhere. Okay, it's better. Chlamydomonas reinhardtii is an experimental organism. So it's used widely in many labs to discover several aspects of the biology and science. Some scientists try to research on the movement of the cilia and the sensory transduction, or like its ability to adaptation to different environment, like light or dark, or high rich nutrition, or like poor nutrition, something like that, and also like photosynthesis. And it's also very hot experimental model for biofuel production, because when it's to do like photosynthesis, the hydrogen and the lipid, it's this green algae's byproducts. It's very useful. And the whole genome of this green algae is about 205 kilo base pair. And comparing to the mouse and the human, and we can see that it's like 10,000, 1 to 10,000. That's the fraction of like a mammalian animal. And it's a single cell, a single cell. It's not animal, but although it can move, it's a single cell organism, and then it's a haploid. That means that it's a kind of its whole genome, like a chromosome, just one copy. Like a mouse and human, we have two copies of a genome. So here it shows that basic structure diagram. And this green algae, it has a cell wall and a plasma membrane, and it has two cilium. One is trans and one is cis. And inside there's a nucleus and a cytosol, and there's a starch like a vascular within the cytosol. And very specialized structure of this green algae is the light spot. The light spot is a very unique, highly functional subcellular part is that it has in this spot it has a carotenoid rich granule layer and sit on a thylakoid membrane, and this thylakoid membrane cannot pass through light, but this granule those vesicles can its function is a kind of like a lens. Then the light coming through, then the light coming like reach to this eye spot, then it will go reflect from this thylakoid membrane, and it's labeled with the green color, and it will be reflects back then to the plasma membrane. And there are channelrhodopsins that's located at the plasma membrane. And this figure is labeled with blue. And this reflected these photons can open these channelrhodopsins. Then the calcium and the proton can flux into the cell, then cause the cell membrane potential to hyperize. Between the plasma membrane and the cell wall of the green algae, there's some vesicles located here, and it's labeled with purple. And in today's lecture, we're majorly talking about the light response behavior that's focused on these eye spots. Since there's a like this like quite of like a measure that light can reflect from this surface, this plane. So this functional is quite like some animals. In some animals, the eye like a cow, like a cat, like a dog. There's a shiny membrane that sits behind the retina. So when the light shining like onto this membrane, and it can like reflects back. So at night, when we see the eye of like a cat and shows so shiny, it's not because that the eyes of the cat can generate light. It just can reflect the light from like a house light, from the moon, from the star, and that can so this bright light is coming from refraction. It's just the same case as in the green algae. So this membrane function is like a mirror. So when we go back to see that the channelrhodopsin is located on the plasma membrane, and between the membrane and the cell wall, there's some vascular body located here. And this carotenoid-rich granule is only located at the eye spot. It's not only located, so this carotenoid is not only important for the eye spot, it's also important for the whole cells with how it gets this green color. So if this like this molecules is being decreased, if they totally gone, then the whole algae will be like a transparent one. There's no color, no green, no red. And if there's a little amount of this carotenoid, there will be a green color in the whole cytosol, but the eye spot will still not be red or orange. So this functional, so it's kind of like the major role of this carotenoid is to make this organism green, the whole body green, and it is stored in this eye spot. But it still have some function like a lens, and when the light reflects from here, and this tiny lens can like focus and enrich, make this light brighter to hit the channelrhodopsin on the plasma membrane that's just on top of this eye spot. The channelrhodopsin's the most sensitive wavelength is between 660 nanometer to 690 nanometer. And so this area is a blue light. And it's between so to compare to the human eyes, it's between the rod and the blue cone. So it's quite blue. The photoresponse by the F spot is to inhabit in a suitable environment for photosynthesis. So although it's like the single cell organism, it's a kind of like a plant that it requires the light to do photosynthesis to enrich the nutrition. So it will move. So if it stay in a very dark area, then if it sends, then it will rotate it like slowly, and until it sends the light beam, then it will move towards the light resource. And this green algae have the two cilium, and depending on the relative location, comparing to a eye spot. So one cilium is called cis, another cilium is called trans. There's a little difference between these two cilium. The trans cilium, if it's a bit stronger. It's much stronger, and with higher frequency than the cis. But when in the intracellular calcium is low, then the majority movement is due by the cis cilium. And when the intracellular calcium is high, then this trans cilium will be stronger than the cis. And so the major movement is induced by the trans cilium. So here in this picture, it shows the photo text. It shows that how this green algae move towards the light resource. And the light resource is at the right up corner. The first step is the light. The green algae is like rotated in with its own axis, and then occasionally or randomly. So for this position, so the light come from here, and it will be reflect back by the green membrane, the thyroid membrane will be reflects back. And there's no light hit the channelrhodopsins on the eye spot. But if it rotated to some direction, then it's being captured by this eye spot, and the light is being reflected by the eye spot to the channelrhodopsin on the membrane. Then the channelrhodopsin will be open, and the proton and the calcium will influx into the cell. Then increase the intracellular calcium concentration. So we mentioned before that the higher concentration of the calcium will cause the transcilium to beat. So the transcilium beats, and this movement will cause this the green algae to turn its movement direction and to move towards the light resource. But this is like the green algae that if it stay in a very dark condition, they will move to a brighter condition. But if there's a very super bright uh, light hit this green algae, then it's called photoshock. So very bright light is actually it's will damage these organisms because very stronger lights, and we can see like a laser or strong light, or strong light like the sun. We cannot see directly sun because we know that it will kill our eye, the photoreceptor in our eye. It's the same condition as this green algae. Very strong light is toxic to this organism. So when it suddenly got a shock, photo shock being captured by the eye spot, then it will undergo a two phase. The phase one, it will suddenly like change the beating like a pattern of the cilium. It will directly the both cilium will like beat in a different way that it can change the direction or move far away from the photoshock location. And then the second phase is like you will relax and change move a little bit, then move away from this light shock location, but the ciliars and the behavior is different from the phase one. And this escape behavior is illuminated by sudden strong light and is dependent on the channelrhodopsin on the cilium. So here it shows that some of these experiments that performed in a dish. When you see that like a medium light intensity, the green algae will prefer light resource is shining from right to left, and the green algae will prefer to go towards the light resource. If the light is stronger, then they prefer to move far away from resource, the light resource. If more stronger, they will just try as much hard as they can, like move away from light resource to find like some shadows to hide. So it's kind of like they prefer to stay in a certain light intensity. But for some like mutations, some they just do not have this strong light avoidance behavior. So more lights that attracts more or this more green algae, and this is a mutant, and this is a rescue one, and it shows that it uh, recover the behavior that escape from the very strong light. And this work is published in PNAS. So the phototaxi, like the white type, they prefer if there's a dark condition and bright condition, but this is not so bright. It's kind of like the most favorable light intensity for this green algae. Then for the white type of green algae, you were moved towards the light resource. However, some strains that got some mutant, it's against opposite behavior. It's called a negative phototactive strain. Its name is AGG1. It just kind of like avoiding the light. So whatever it's like more favor the light or avoiding the light, they are all channelrhodopsin dependent. If knock out both of the channelrhodopsins, then the green algae will totally lost this photoresponse. And the cilium movement is also calcium dependent. If it's in a calcium-free condition, there's a totally no calcium outside, and of course there's no because the inside calcium is coming from the outside. So if you take out the outside, generally the inside calcium was also being loosed. If there's a no calcium, zero calcium, the function of the cilia were just dysfunctional. It's not working as so good as normal. It's a little kind of like the cramp that happened in in our own legs. And this is because the cilia's movement is dependent on the calcium. So lower calcium or higher calcium concentration will change the movement behavior of the cilia body. And this is a diagram that shows that after there's a light shine on this eye spot, it's focused by those orange like the carotenoid vascular and focused on channelrhodopsin 1 or channelrhodopsin 2. And this influx of proton or sodium or calcium can cause the membrane potential to depolarize. And this depolarized membrane potential will open voltage gated calcium channel. And the calcium influx will change the behavior of the cilium. And there's another very interesting part is that if there's a low calcium, the angle between these two cilium is also different. If the low calcium, the angle is larger, and if on high calcium, the angle will be smaller, will be more sharp. So this also can explain why in the photoshock the whole cilium becomes very straight and moving like far away from the light shock location. And it's also because that the angle of these two ciliums is calcium dependent. And so that's all. Thank you.